My name is Medha Lolayekar, and I'd like to present to you a brief summary of my project, Identification of Putative Inhibitors Affecting the Regulatory Regions of Selected Genes Involved in AML.
What is AML? AML, or acute myeloid leukemia, is a fast-progressing disease where myeloid cells rapidly grow, interfering with the production of normal white blood cells, red blood cells, and platelets. The main cause of AML is a genetic change in the DNA sequence of hematopoietic stem cells or myeloid cells. So what exactly happens to the blood during AML? The overproduction of immature myeloid blast cells takes up space in the bone marrow and thus killing the healthy blood cells and platelets. My project was to recognize certain genes that cause the manifestation of AML. I researched the gene's direct binding and whether it was upstream or downstream of the target gene. I would then look for inhibitors that could be potentially used to inhibit the activity and in turn prevent AML from further developing. The objective of my research was the identification of regulatory regions associated with certain genes which are important for the manifestation of AML, and the identification of novel inhibitors which could be found on regulatory regions of target genes. My workflow, I explored genes related to AML using the cBioportal software and NCBI software. Using the data from these softwares, I focused on a few genes to study for my research. I utilized the cBioportal datasite, which provides different kinds of samples and cohorts for different studies on AML. And based on mutations and most prevalent genes, I selected a few genes for analysis. Then, I researched cell lines and pathways that connected the chosen genes together. Lastly, I looked for an inhibitor that could target the genes to limit the proliferation of immature myeloid cells. And lastly, I present my findings. Here's a list of some of the most prevalent genes in AML and a pie chart comparing them all together. Again, I went to the KEGG database and found a pathway where AML1 or RUNX1 directly binds to the CEBPA gene. Through this pathway, I looked into RUNX1 and its downstream to CEBPA. This leads me to my result. CEBPA causes mass proliferation of myeloid cells in the bone marrow. There's no inhibitor that directly binds to CEBPA, thus through RUNX1, we can inhibit the expression of CEBPA. My identification of a novel inhibitor included the BET protein, which is still in testing but could be used. To end off my presentation, I'd like to summarize my findings. And so for the discussion, I found direct binding of RUNX1 to CEBPA. I found a cell line which has positive expressions for all three genes. I found one inhibitor known for RUNX1. And in the future, I'd like to test the activity of this inhibitor in the cell line. We can block the activity of RUNX1 using this inhibitor. Thank you.